So I'm just continuing the PowerPoint here. My Zoom and internet failed, so lost a good bit, and I'm just resuming here. So for twin studies, in identical twin studies, you have identical genotypes. That is, their chromosomes and genes that they contain would be identical. But in contrast, the genetic similarity between zygotic twins, fraternal twins, is on the average 50%. So in twin studies, investigators would study records to identify pairs of twins in which at least one member has the trait, for example, a diagnosis of a particular mental disorder. If both twins have been diagnosed with this disorder, they're concordant. If only one has received this diagnosis, the twins are said to be discordant. Adoption studies require that the investigator know the identity of the parents of the people being studied and be able to measure the behavioral trait in the biological and adoptive parents. If the people being studied strongly resemble their biological parents, we conclude that the trait is probably influenced by genetic factors. To be certain, we will have to rule out possible differences in the prenatal environment of the adopted children. So for example, in pair one, they might have the same DNA and the same environment. So if intelligence is different, we can assume that there are tiny differences in the environment, like, for example, their friends, the time spent studying, that could cause the difference. In pair two, you have the same DNA and the same environment. However, the twins often grow up in very similar environments. So if the intelligence is the same, it's hard to tell if it's due to genetics or the environment. In pair three, you have the same DNA and a different environment. So now the twins are adopted by different families, but grow up in different environments. If the intelligence is the same, it is likely due to genetics. If intelligence is different, uh, if on pair four you have different DNA in the same environment. So here, if one sibling is adopted, here one sibling is adopted. So if intelligence is the same, it is likely due to the environment. If intelligence is different, it is likely due to genes. Adoption studies require that the investigator know. Oh, right. So in genomic studies, it's important to understand the concepts of genome and allele. A genome is a complete set of genes that compose the DNA of a particular species. Alleles are the nature of the particular sequence of base pairs of DNA that constitutes a gene. For example, the genes that code for blue or brown iris pigments are different alleles of a particular gene. Several longitudinal studies in the 1970s and 1980s found through a correlational analysis that smoking cannabis before the age of 15 increases the risk of schizophrenia from 3% to 10% by age 26. But why would smoking cannabis raise the risk for some but not all people? This first study here, published in 1987, was from data obtained some time ago in the Swedish army. At the time, not too long ago, it was required that all male members of Sweden serve a period of time in their military. So they just had all of these data laying around and, had some follow-up studies on this population after the fact. And, this wasn't actually a hypothesis-driven question. It was all done post hoc after the data had already been collected. So a group from Britain, led by Robin Murray, amongst others, went down to Dunedin, New Zealand, this area where people also tend not to leave, and then there is also a lot of cannabis use there. And really, they took part of a larger longitudinal study, but in their little small part of it, they looked at this one gene that encodes COMT. This is an enzyme, much like MAO, that breaks down the catecholamines, dopamine, epinephrine, and norepinephrine. So COMT is particularly important for the breakdown of dopamine in the frontal cortex. The main reason people say that is because there aren't as many dopamine transporters up there. But regardless, a functional polymorphism of the COMT gene results in two common variants, the valine and the methionine allele. And what has been shown is that increased COMT activity associated with the VAL allele may result in a combination of reduced dopamine transmission in the frontal cortex, leading to potentially an increased level of dopamine signaling and psychosis. And it is just this allele that was found to have an increased risk for schizophrenia, just this one allelic phenotype, when they looked at the relationship between COMT and cannabis use in amongst people in New Zealand. It's also worth pointing out that relationship, that study hasn't been replicated everywhere it's been conducted. But when it was replicated in New Zealand, they did find out or uncover this genetic relationship as well. So only if you had this one unique allelic phenotype of COMT would smoking cannabis in adolescence lead to psychosis in adulthood. And again, I wouldn't put too much faith into that one study, just know that that observation has been made. Your book mentions many research methods to study the underlying physiology of the brain, but what about behavior? That is sort of a key component of biological psychology. So when we do these manipulations like an ablation or perform optogenetics, we often are doing them during a behavioral assessment that can be quantified in some empirical fashion. So the techniques that I'm going to talk about are condition place preference, operant behavior, IV drug self-administration, the Morris-Water maze, and the elevated plus maze. Condition place preference is a very raw, sort of crude, fast throughput method to obtain data on whether or not a drug produces rewarding or aversive effects. So what you do is you put your animal in a oftentimes biased chamber like this, where one side looks or smells one way and the other the opposite. And if the animal spends more time in the environment associated with a drug, it would be said that it produces rewarding effects. If they then, to avoid the side that was given a drug, they would say that it produces aversive effects. During operant conditioning, the animal responds to actually receive some sort of reinforcer. A reinforcer would be an event that is maintaining the animal's operant behavior. If the animal is receiving something when they press a lever, for example, in a cage, that would be an example of positive reinforcement. In an earlier lecture, I showed you a video of one of my animals responding for electric currents delivered directly to the brain. That would be an example of positive reinforcement. Negative reinforcement is also something I've used a lot in my lab. Negative reinforcement is where the animal increases responding to avoid a stimulus. I've tended to use electrical shock, but there are many examples of this in, maintaining or modifying animal behavior, where you might want to see more A's. For example, you might want to see your child make more A's, and therefore you would add in something that they could avoid if they continue making A's. Like, if they continue making A's, they don't have to wash the toilet that week, for example. IV drug self-administration is an adaptation of operant behavior. This is commonly assesses an animal pressing a lever to receive IV injections of a drug. Typically, I implant the jugular catheter into the jugular vein and then snake it around so that it is leaving the animal between the shoulder blades. And then that line would be connected to a pump. So every time the animal presses the lever, the pump would turn on for a fixed amount of time, delivering a fixed amount of an IV drug injection. To assess memory, we typically use the Morris water maze. This involves a circular pool of water it's made opaque by the addition of milk or a dye. This really stinks after a few days, let me tell you. The animals must swim until they can find the escape platform that's hidden just beneath the water. In terms of spatial memory, there are typically signs placed outside. Like, for example, that's what you can see this red and white checkerboard and the yellow triangle are. Those are contextual cues that the animal can use to remember where the platform would be. And the animal would learn the spatial position on the platform. And then, for example, you can give them cannabis and watch them take much longer to find that same platform because it impairs spatial memory. Here is a video of an animal behaving in the Morris water maze. So that is one way of assessing spatial memory. Also, working memory. Although there are different ways of getting at that, including delayed nudge match to sample. I guess spatial memory is really what the Morris water maze is best at. But how do you get at anxiety? So again, we have many measures of that, but one of the common and simple ones is known as the elevated plus maze. So here you have an elevated plus maze with two closed arms and two open arms. By that I mean two arms without walls and two arms with walls. As you might imagine, if you've done much hiking up in the mountains, it requires less anxiety if you are walking out on a limb or a ledge in which there is a wall or a bar. If it is wide open, that tends to increase anxiety. So if a drug produces anxiety, then they would spend less time on the open arms. if the drug produces anxiolytic effects, meaning it relieves anxiety, the animal will spend more time on the open arms. Here's what that looks like. The green arm is clearly open. And that, is it for animal behavior. So, I've done all of these techniques and many more. I can go on and on about these, but I don't see the, need to belabor a lot of the details of these unnecessarily. So, right, this is what I'm going to test you on, and feel free to email me any follow-up questions.